See the genome like never before. Irys allows you to visualize long DNA molecules for a deeper understanding of genomic structure and function. Maps are essential in everyday life. When you consult one, you need to see more than the street you're on. You need to know where that road goes and which roads lead to your destination. Without context, all streets look the same, and one neighborhood looks like the next. You need a map that tells you where you truly are. Only when you zoom out do you get a more complete map. You see how roads connect, which turns to make, and where your destination lies. That's a map that can take you places, help you recognize landmarks, and find locations of interest. You're not just on any street. You're in a city on the west coast of the United States. The same is true in biology. Context is essential. Irys gives you a genome map that broadens your horizons far beyond single base pairs. Only when you zoom out do you see a more complete map. Now you see the architecture of the whole genome in a single view. Genome-wide order and orientation become clear, allowing you to directly see connections between elements and detect structural variations such as translocations, repeats, and deletions. Irys delivers this more complete genome. To get started, simply extract DNA molecules, several hundreds of kilobases long, and follow the labeling protocol. Endonucleases in the labeling kit create nicks at 7 base recognition sites across the genome. Fluorescent nucleotides are then incorporated to generate a unique sequence-specific pattern. Long DNA remains intact throughout the process, preserving architecture for direct observation. Pipette your sample into the IrysChip and place in the instrument. Inside the chip, molecules are suspended in solution and directed through an intricate field of microfabricated pillars to unravel coils. After entering nanochannels, the fully linearized molecules are imaged, revealing their fluorescent label signatures. Scanning molecules in this way across thousands of parallel nanochannels and over several cycles per sample, the high-throughput Irys generates enough data to create an accurate map of the entire genome. The raw image shows fluorescent labels on each molecule as dots on a string. Using those signature patterns from individual molecules as reference points, the system overlays images to build a complete consensus map. Armed with this, you can examine DNA at single molecule resolution. To identify structural variation, simply compare your genome map against a map from another individual or a reference genome. Or you can use it to direct de novo assembly of next-gen contigs. The possibilities are endless. Bring new insight to your genomic research with Irys from BioNano Genomics. Altogether better.